All right, hello everybody. Welcome to Six Harbors Brewery when we're celebrating Movember. November is Movember, Men's Health Awareness Month. Hello Facebook Live, hello everybody here. Facebook Live, you can't see it, but they all have fantastic mustaches. Today I'm going to be introducing you to Dr. Lloyd Trotman. Dr. Trotman, Professor Trotman here at Cold Spring Harbor Laboratory. He got his PhD from the University of Zurich. And then a little bit about 6 years later, he came and joined Cold Spring Harbor Laboratory. Lloyd is an avid surfer. So if you're wondering what the big waves of Long Island are like, he's the guy to tell you about it. Lloyd's even more of an expert when it comes to prostate cancer, particularly prostate cancer that is unusually deadly. So he's going to tell you a lot about that tonight, a lot of other things, and if you have questions right afterwards, that's great. A reminder, if you didn't know, every beer that's bought tonight is $1 donated to the lab for cancer research just like Lloyd's and other research. So thank you, Six Harbor Brewery, for that. Yeah. All right, so without further ado, I'm going to introduce you to Dr. Lloyd Trotman, or he'll introduce himself. I don't know what I'm saying. So I'm going to give you this.
Okay, so we can flip it on. Let's put that on there. Great, thanks. All right, can you hear me with the audio? Is it yes? I can hear myself now. I don't know if that's good. All right, so the sound is good in the back? Great. Thanks everybody for coming out. It's really exciting to be here, to see this venue. I usually go to the brewery in Oyster Bay, but I might have to reconsider this. Very sweet spot here. So, first thing I have to say is, it's really great that everybody came out, and I'm actually unusually nervous today because I think this is the first time I'm actually giving a talk in the presence of my wife, which is sitting back there with a very unusual mustache. So forgive me if I stumble. And so the second thing, there's also some team members here, and that's actually really great because you can ask people who do the work that I'm trying to piece together about what is actually going on. So there's Grinu I'm seeing in the back with Hao Tian. Grinu came to the lab a few years ago, and I'll tell you some of the things that she's working on. Then we have Hillary somewhere. Yes, right here, front row. So she's also been in the lab for a few years. So here are some real experts on what we're actually doing. So the first thing that I wanted to start out with is you've been wearing the mustaches. I'm growing a mustache. I don't usually have this mustache. It's actually going to go away December 1st because it's on for November. Okay, yes. Second thing, I'm geared up with the Prostate Cancer Foundation, a fleece jacket. The Prostate Cancer Foundation is probably the most important organization bringing prostate cancer scientists together. So they're extremely helpful in advancing this research. And the third thing is obviously the brewery, the beer here. So thanks for hosting this as well. So what I wanted to start out with is give kind of an overview of why one would do discovery research in cancer when cancer is such a acute problem, and you might expect to find researchers who are also clinicians doing the lion's share of that work. And so by means of introduction, I have a few questions for you. So the first question is essentially, does anybody or can anybody guess? You all have heard of Lance Armstrong, famous cyclist. He has won, I think, the Tour de France 7 times, right? And so if I ask you what the drug is that allowed him to win those 7 titles, what would you say? EPO. Erythropoietin? Anything else? Steroids? Well, both of those are wrong. That's not why he won those titles. The reason why he won those titles is actually a drug called cisplatinum. Cisplatinum saved his life because in 1996 he was diagnosed with testicular cancer, an aggressive variant that's actually metastatic and would have killed him within just a few years. And thanks to cisplatinum, he survived. And then could go on to greater things. So yes, that's his story. But so what's cisplatinum? Cisplatinum is the result of about 20 years of accidental things. So the whole story starts, and this is why I want to tell you about it, with in the 60s, when scientists were really interested in how cells could be killed, because it was getting clear that this could be very important for cancer research. Many different scientists tried many different things. And so some scientists that you wouldn't bet a lot of money on had the idea that when cells divide, and you're looking at the picture of the DNA, the chromosomes that are all nicely lined up, and they're being pulled apart by what we call a spindle inside the cell. When you look at that image, it looks a lot to a physicist like an electric field or electric field lines between two poles. And so very few scientists, actually, probably just a handful, thought this is all electric. And so they devised some experiments to test this hypothesis. And every sane scientist could just laugh about this, obviously. The experiment was even more laughable that they designed. They actually started out with some bacteria in a broth and placed 2 electrodes into that broth. Okay? That research was not funded. But it gave them a very clear result that, again, nobody would think has anything to do with cancer. Essentially what they found is that the bacteria in the broth clearly changed their shape. They became extremely long, extremely elongated. Okay, so every reasonable funding agency at that stage would tell them that, you know, this is nice, but let's move on to, you know, higher objectives here. But they, being actually was a bunch of chemists and physicists, they thought that we've done something and there's a clear effect, let's find out what happened. And thanks to their persistence in trying to find out what happened, 10 to 15 years later, it became clear that there is a compound that is released from the platinum electrodes that they used that actually kills those or arrests those bacteria. The compound later was identified as cisplatinum, which is extremely hard to make and describe, so it took very long. And then at some point they actually then did animal experiments, and the next thing was, I think that's about 20 years later, they did experiments, University of Rochester, I think, did the first experiments on people who had testicular cancer and cured about half of them. And so it got FDA approved. So this is the first vignette for how breakthroughs actually happen. You know, when we're thinking of breakthroughs, we're usually hearing about the tip of the iceberg, the very last few experiments, the experiment that goes into the clinic, but we don't hear about all the craziness that's behind that. Another example that you've probably heard about a lot recently is the success story of immune therapy, which after not having been taken super seriously by cancer researchers, all of a sudden has seen a breakthrough in the last, let's say, 10 years. And a third example that I want to give is about prostate cancer. And it's also very interesting because it's the mainstay of successful prostate cancer therapy today is hormone therapy or anti-hormone therapy. And if you look at how that actually came about, it's again very, very illustrative and very interesting. So what actually happened is that a scientist in Chicago was studying the prostate, normal prostate. And in order to study that, he used dogs. They have prostates. And the question that he asked was whether that gland, in terms of its function, is in any way responsive to hormones, such as testosterone. So he would castrate dogs or not castrate them and find out how the prostate reacts. And so that was perfect research, and he showed that the prostate gland was dependent on those hormones and so on. These studies had nothing to do in concept with prostate cancer. The only reason why he ended up making seminal discoveries for prostate cancer was that by chance, dogs are among the few animals that can naturally develop prostate cancer. So among the animals that do develop prostate cancer, there's us, there's dogs, and it's also known that lions can develop prostate cancer, okay? And so he was, quote unquote, lucky enough, but obviously he thought it was a problem, that every once in a while when he was looking at a prostate and the reaction to the hormone withdrawal after castration, he would actually have a dog that had a cancer. And that was, of course, annoying because it messes up the study. You want to have normal prostate, and he was studying the effect of normal prostate. And it was also clear at the time that a cancer is completely independent and different and has evolved away completely from the tissue of origin. It's a different beast. So he would have never asked the question whether the dependency that the normal tissue has would also affect the cancerous tissue, because they were thought to be two completely different things. However, he saw that the reality was different. He saw that dogs that had actual cancers also saw their cancers shrink. And that was the beginning of what has 60 years later still remained the standard of care therapy in prostate cancer, hormone therapy, anti-hormone therapy, hormone withdrawal therapy. So that example, the immunotherapy, and also the example of the cisplatinum, they highlight essentially the fact that people who haven't necessarily been asking specific cancer questions have made seminal contributions to the best ways of fighting cancer today. But they also highlight another aspect, and that's where we come in. The example of the dog is interesting because it completely translated to human. So what they found in dogs was completely applicable to human as well. The second example, the immunotherapy, was the discovery in mouse. Jim Allison developed everything that we now know about additional keys and locks on immune cells that tell an immune cell to engage or not engage. He found all of those in mouse, and he defined them all in mouse, and it translated perfectly to human. The third example was bacteria. Bacteria went to mice, mice went to human. And so we as cancer researchers have been thinking along the same lines. It is very hard to study the things we want to study and to engage a system as critically as we would like to study it when we're looking at human. It's much easier if we're doing it in a model organism. And so we chose mouse. And with that, there was only one problem, and that is, as I told you before, mice don't get prostate cancer. So our first challenge was to actually make mice that get prostate cancer. And it took us quite some time, but I would say about 5 years ago or 6 years ago, we were actually able to develop a mouse that gets prostate cancer, and it doesn't just get prostate cancer, it develops metastatic prostate cancer, and it will actually die of that disease. And without us taking any human cells or doing any manipulation and sticking them into the mouse, it's the mouse with its own disease dying of that disease. And on top of that, we were actually able to get the disease to light up so that we can see it through the skin with super sensitive cameras. And so we've put ourselves in the position to have the third mammal that actually gets prostate cancer, well, with our, with some tricks, but then also an animal that we can study the disease course of and in. And so that's kept us busy for the past 5-6 years, essentially. With these animals, we're trying to do two completely separate things. And the first thing that we're trying to do is to understand and answer essentially the basic and big conundrum of prostate cancer, which is that prostate cancer is very common. When it was first identified, it was described as an extremely rare disease. Today, something like 1 in 6 men will have a lifetime risk of developing prostate cancer, so it's very, very frequent. And the only difference between 150 years ago when it was first described and today is our lifespan. Our lifespan has essentially doubled over that time. And that's why now there are all these cases. So with this disease now, the amazing thing is, and that's one thing that separates prostate cancer from many other cancers, this thing is that having the cancer is actually not a problem. And this is a bit hard to wrap our heads around. The vast majority of prostate cancers remain indolent and will never be life-threatening. So that's the good news about being diagnosed with the disease. The problem is that there's about 10 to 15% that actually will be more aggressive and will become therapy resistant. And to this day, it's very hard to separate the two. The standards that are used today in clinics go back again about 50, 60 years, and it's essentially looking at the disease and looking at the shape of the disease. It requires extremely trained eyes to do that, and that's still the best predictor of how somebody's going to do over the next 10 years. It's better than anything molecular or genetic that can be done today, which is pretty amazing after all the genetics revolution. So we're thinking that with our own mouse patients now at Cold Spring Harbor, we should be able to study this in greater detail. In fact, our mice, even though we engineer them to develop lethal disease, they don't all get it. It's something between 50 or so % that do not get the lethal form of the disease. And at first we thought, well, we'd like to have every mouse get the same thing. But then we realized, well, wait a minute. If it's exactly the problem in clinics to separate the ones who don't get lethal disease from the ones who actually have metastatic, and now we have a model that reflects that same difference, well, why don't we go ahead and study it exactly in our model? And so that's what we've been doing for a long time now. And I think one of the breakthroughs that has happened is that Greenu, who is here today, she has been able to isolate cells from these animals long before they metastasize. And we've been able to realize that in the prostate even, they come in two flavors. There's the kind that sits and grows. And there's another kind that grows, but it doesn't sit. It migrates. And so she's been able to isolate cells and culture them in dishes. And that's quite a feat, because I would say until, well, there's essentially only one successful example of a lab having done that. And so she's been able to isolate those cells, and lo and behold, when we look at cells in the dish, they also come in 2 flavors, and they maintain those 2 flavors. They either sit and proliferate, or they migrate and proliferate. And so now we have a really great tool to actually understand the differences between those 2 cell types. And that's what we're spending a lot of time on. The hope is that we're finding things that are fundamentally different between them that we can then test patients for and answer essentially the question whether they're more likely to develop the lethal form of the disease within the next few years. So that's a major thing that we're engaging in. There's lots of tools that we can use. We can use CRISPR technology, we can use sequencing. There's a lot of recent emergent technology to really help us get at that question. The second vignette that I want to give is about another question that we're able to answer now or to address now with those mice. And that is, again, it's completely different. In that one, we're not trying to describe what's going on and what the difference is. But I told you that we've been able to get this disease to go metastatic and kill a mouse, but to also see it as it's doing it. And that's a technology that's been developed a few, let's say 10 years ago. It's using a gene from the firefly that is allowing fireflies to communicate and shine light. It's using that gene, and we're getting it into the prostate. And so that light can actually be picked up with super sensitive cameras so that you can tell whether the disease is still in the prostate or whether it has gone to different places. And so we can monitor our mice as they progress with the disease. And so obviously then our question is, well, are there new, better therapies that could block disease progression? And while most of the people who are in a hospital-style environment would now ask the question, well, there's hormone therapy. How can we improve hormone therapy? What kind of developments are there that get harder at hormone therapy, second line, third line, and so on? We're not doing that. We're somehow thinking that this is really good and interesting, but it might not get at the core of the disease. And there's one thing that you'll hardly hear discussed or addressed in that setting, and that is essentially that the largest discrepancies in prostate cancer lethality are seen on a global map, essentially. When you look at a map of the Earth, and you're looking at regions and lethality of prostate cancer, you realize that Western Europe and the US are among the highest ranking. And you also realize that Asia essentially hardly knows lethality of prostate cancer. In fact, many Americans and Europeans who live in Asia kind of complain about that because they're a bit lonely when they're in the ward. It's not the general awareness. It's not a problem essentially. Lethality is about 10 times less than what we have here, and so there's obvious questions as to why that is. And there's a very interesting phenomenon that's been studied quite seriously, and that is that when Asians, or particularly Japanese, who have a very low death rate of prostate cancer, when they move to the US, their death rate increases. So it sounds like it's not genetic. It's somehow environmental, as far as one can tell from these studies. These are not so easy to do in a controlled fashion, but that's what the epidemiological data essentially say. So it sounds like we should be studying lifestyles and diets a bit more. And we were thinking at the time that this is something that we could be doing with these animals. However, we didn't want to just say, look, let's have these animals exercise more and, they'll be doing, because the answer is going to be they'll do better, and then how, it's like you can't convince anybody to change their lifestyle, in that way. We wanted to get at actual molecules, agents that could change the outcome of the disease, because we know our mice are all potentially on a life-threatening course. So what we've done is a look at one large-scale study on diet that has been done in the US. And that study was called SELECT. It used over 30,000 men, and it asked the simple question whether antioxidant supplements, whether they have an effect on prostate cancer. And the answer was a resounding yes, in the exact opposite way of what everybody was hoping. Everybody thought that antioxidants are great. Antioxidants are exactly what you need, because oxidants are equal aging and damage and you name it. Antioxidants keep you young. And the exact opposite is true, at least for prostate cancer, some other cancers as well. So there's an approximate lifetime risk increase of almost 20% for men who took antioxidants, selenium, or most prominently vitamin E, on a daily basis. And so with that data in hand, we were thinking, well, if the antioxidants are bad, could it be that oxidants are actually good? And this hasn't been tested. In fact, actually, the people who, 30,000 men, you can imagine, that's one of the largest studies on prostate cancer. And it's extremely expensive, so it's become hard to convince people to do similarly large studies without having a strong winner at the end, right? And so we've asked in cohorts of mice whether oxidants could block or delay the cancer. And so we've done it with a combination of two agents that can be increasing oxidative stress in cells. And one of those agents is a derivative of vitamin K, and the other one is vitamin C. And essentially what we've been seeing is that generally the oxidative stress actually suppresses the development of the cancer very nicely. I mean, we were actually surprised. And the only thing that this left us, it was interesting to see that the vitamin C per se doesn't have a big effect. So the vitamin C is thought to be an antioxidant, but there's data to suggest that it actually can work in an oxidant fashion. However, in mice, they produce their own vitamin C, unlike us. So we're a bit unique in mammals that we're not producing vitamin C. So it's we don't even think that giving them vitamin C orally had any effect at all. Other people have shown that when you inject vitamin C, that you do see some effects. Again, oxidative stress. But the vitamin K derivative very strongly showed an effect on its own. And so Manny is there in the back. So he's Mr. K. So he's been fortunate or unfortunate to try to come exactly at the time when the question was asked, well, how does this work? And so he's behind all the headway we've made into trying to understand how this actually works. And the interesting thing is that obviously if you went through literature, everybody already knew how it works. It's just all wrong. So it's been extremely exciting for us to actually try and figure this one out. And the hope is that at the end of the day, we could be doing things that are very simple to reduce the risk of prostate cancer, just like vitamin E would increase the risk, but then also measure how people are doing and whether they're taking enough, whether they should be doing some more, and whether it could be combined with other things that go in the same direction. And this is something that we now, Cold Spring Harbor is partnering with Northwell Health, so we can be doing with our friends at Northwell Health. So those are the two vignettes essentially that I want to share from the lab. I'm happy to answer more questions if there are any. There are other things that we're doing that are ready or not ready for prime time, but I'm happy to talk about more of that.
Yeah, you know what I'm going to do is if the front row you can feel free to talk directly to Lloyd, and back row, I'll be drifting around.
All right, great.
Yeah.
Thanks. Yeah.
Yeah, you know, question in the back right now.
Two things. Many years ago, I was at one of the lectures for breast cancer.
Yeah.
And I asked the scientist, doctor, what animal had a similar breast cancer to female or Homo sapiens or people? And he said dogs.
Okay.
And I was amazed when I heard you tonight talk about prostate cancer, and dogs were the closest similarity. And number two, when cancer forms, the cells keep dividing and growing. Why does the body not recognize it as a foreign object and go to destroy it?
Yes.
Or fight against it?
Yeah. So today there's essentially two answers to that. The first one is because it's your own cells, so they aren't foreign, and the second one is because the cancer actually evolves immune suppression. If it doesn't do that, it gets attacked. So there's a selection against those who are attacked, and for those who can go under the radar. And so the immune therapy success today really is in the realization of how they evade, how they go undetected, and presenting them again. Unfortunately, this hasn't worked well in prostate. It's worked quite well for skin cancers, melanoma particularly. And it has, for the first time, given essentially lifelong responders that just instead of having a high rate of people who respond well to therapy, it may be a low rate, but they're protected forever, just like immunization usually will protect you. So it's a completely different paradigm from trying to kill the cell with drug to have the immune system attack. Yeah. So that's a lot of prostate cancer research is trying to make the prostate cancer very immune attractive.
So two other thoughts is, I'm sure they're thinking about it, but my question is, if you have a tumor and it's obviously your own cell, is there a way to inject foreign cells
Yes.
into it so the body will recognize
Yes.
it as an alien?
Yes.
Yes.
Yes, there are actual therapies like that. Sloan Kettering is doing some of those where cells are taken out, they are actually modified and put back in. Yes, those are complicated, but people are going in that direction. Yes, exactly.
Lloyd, another question back to you.
Yes, sure.
How accurate are the results when you've taken another species and
Yes.
manipulated that species to have cancer?
Yes, yes. So, as mentioned, you can only generalize because you don't know for your specific question how well it translates. Right? We were the other day actually looking at something, a gene that we're really interested in, that we've studied in human cells, and then we realized that the mouse gene is lacking some parts that we're really interested in that gene. So there we couldn't do any parallels, right? We can't use the mouse essentially for that question. I would say what drives us, I mean, we've had the example of the dog translating to human. The immune principle of essentially how the immune cell decides whether to attack or not attack, was really extremely well conserved. So the guy who got the Nobel Prize, or the two who got the Nobel Prize a year or two ago, could take their discoveries and try them right away in human. And the more mechanistically they knew, the better they could design drugs for humans. So really great translation. What keeps our hopes high for prostate is the following. We know that essentially 2 genes are among the most consistently mutant in human lethal prostate cancer. When we use those 2 genes, that's how we make our model. And it's not like we somehow ask those cells, to do exactly what they do in the human disease, meaning leave the prostate, settle in bone, in liver, in lung, and kill the host. They just do it. So we're thinking there's a fundamental program that's conserved in biology, which is a crazy program. I mean, why would a cell, do that, right? But apparently it's conserved, so our hopes are high that we can target it the same way.
The beauty of Long Island around July the 4th are the random flashes in the night.
Yes.
As the firefly emerges.
Yes.
Could we use that observation to randomly insert it into the genome to light up the situation, back sequence the clues?
So essentially you'd like to make tumors that flash?
No. You have your model, and
Yeah.
you've been following that
Yeah.
with the firefly.
Yes. Gene.
But if we now randomly put that in, it could interrupt a clue.
Yes. Yes. That's a technology that's been used extensively. It's actually called Sleeping Beauty. It is derived from a discovery that was actually made at Cold Spring Harbor and gave Barbara McClintock her Nobel Prize, the discovery of jumping genes that move around in the genome. And so some scientists thought, okay, exactly like you just said, if we have a gene that disrupts things, randomly move around in the genome, can we then find out when it's disrupting something that's really important for a process? Yes, that is an approach that can be done. We haven't used it in our line. What we've been thinking is that when our cells, the cells that start in the prostate and move somewhere, they might randomly accumulate those changes. And so we've hired your wife, essentially, to help us sequence all those changes and find out what's actually going on. And so that's one of the things that we're doing. And I think it's been one of the major clues behind finding out how the vitamin K actually works. Yeah.
You mentioned that you try to identify the cells that sit there and grow versus the ones that spread.
Yeah.
And it takes expertise, and I assume it's done visually, right? I was wondering if you could make use of advanced machine vision technologies with deep learning and artificial intelligence to teach a machine to identify those cells and help you out.
Okay, so the hard part in that one was, in general, to get cells out of the mouse and keep them alive. That was the hardest by far. And in fact, it's been impossible for human cells, for example. It's been pretty much impossible for most cells, cancers or not. In fact, prostate cancer has remained an extremely hard cell type to culture. Today, there are something like 100 or more than 100 lung cancer cell lines that people can study. There's only about a handful of prostate cancer cell lines that people can study. So about 5 years ago, a group in Holland started to develop the key breakthroughs that actually allowed everybody to take cells into culture. It's still very hard, but that was the hardest part, and we can use that technology now to get those cells. funny enough, the migration versus non-migration is black and white. We don't need machines. Essentially, we don't need machines to see it. We need machines to quantify it, to track cells and say, okay, so on average, per minute, this cell is moving that much and the other one is not. So we can actually do assays and statistically say, you know, we've changed from moving to non-moving and so on. But it's not, this assay that we have today is so black and white that we don't need really advanced technology to get to see it.
So what do the expert humans?
Sorry?
What do the expert humans have to? What are they trying to do?
The human.
You said you needed expert humans, like to identify, distinguish between two things.
Oh yes. Okay, now I understand. Yes, yes, exactly. So you are right. In human, you don't see live cells. You see them fixed. And there, you need the pathologist, exactly, that's extremely expert, and so on. And there, Sloan Kettering, for example, I know because they've been talking about it and then getting in trouble about it, they have developed software to exactly do that, machine learning and so on and so forth. They've just not been so clear about this to patients. That was the problem. They've used a lot of the problem that they have with your approach is the difference between Google finding out if your picture has a dog and Sloan Kettering finding out if that prostate has a tumor that's going to go indolent. There's a billion dog pictures on the internet, and it's easy for anybody to step in and say, no dog, yes dog, no dog. There's only tens of thousands, hundreds of thousands of pictures of tumors that will go bad, and there's only a handful of people, so to speak, to say this is right, this was wrong. So it's a very laborious. You need mass, you need numbers for the machine learning, and the numbers aren't yet there, but they're trying. So that's one of the things. Our hope is that they're barking up the wrong tree because we're hoping that there's something underneath that's just manifesting itself in that shape, but that's biologically clean and clear. That's the hope, that separates those two. Yeah.
We're going to circle back to the back here.
Okay.
Hi, I have two questions. One is there a relationship between benign prostatic hyperplasia, whether they're more likely or less likely to get prostatic cancer? And the second question is, I read recently that, and I don't know if this is new news or old news, but to me it was new, that they found that cells can actually communicate with one another through mitochondrial DNA that they transfer between each other, and actually you can take, for example, viral RNA and transfer it to another cell. Is that somehow that it's significant the way a cancer might spread?
Okay, so to go for the first question, to my knowledge, there isn't really a correlation between the hyperplasia, the benign hyperplasia, and the tumor. When we first started modeling all of these with genes going wrong and so on, we got a lot of hyperplasias that really never made it to the tumor stage and were very frustrating. And we realized that there's a lot more that needs to happen to actually give a cell a clear advantage and take over. So regarding the second question, there's quite a bit of recent evidence and interest in the transfer of material between cells. There are quite a few very astonishing reports in that respect, including cells taking DNA, cancer cells essentially taking DNA, packaging it, moving it out, and then that being picked up elsewhere and essentially being bad for the environment there, meaning the cancer cell having an advantage of finding that spot and essentially preparing the soil for a later invasion and metastasis. However, the field is really, really very evolving. I think it's still very early days. There is one aspect of it that I sense is going to have long legs and being quite rewarding, and that is we still have the problem that to essentially tell somebody how bad their cancer is, we have to see it. And we wouldn't really want to do that, because we also know that every biopsy that you take comes with the penalty of inflammation and things like that, and that's the last thing you want. And so being able to remotely tell whether a tumor is doing well or is well behaved, especially in prostate cancer, or whether it's getting bad, would be really great. And so people are looking at blood. And since we can sequence anything under the sun, everybody's looking whether they can sequence and find DNA that says things are going to go bad, including us. And so the one thing that we've been looking at is, in fact, particles that cells use to communicate with each other, because they are enriched in blood. And they are enriched in things that come from a tumor. And so it's ongoing research. I think the field I think some dust will settle, and some things might be a bit too sensationalist now, but others might really pan out. Yeah.
Do we have any questions in the front that I can pass this to? No? Okay, I'm going to move back to the back. Yep, I knew I had one over here.
Two-part. Earlier you had said that many people have cancer, but it's suppressed. What was that percentage?
So something like 80% of prostate cancer, people diagnosed with prostate cancer, will live with their prostate cancer, not die of that prostate cancer. They're saying they will die with that cancer.
How far back can you track cancer? In mummies or whatever? How far back can you actually say the population cancer? 100,000
Yes.
years, 2,000
Yes.
years?
Yes. So the book to read on that, that's essentially the first chapter of Siddhartha Mukherjee's book, The Emperor of All Maladies. It's an amazing book. I really, it's the first time somebody's written a history of cancer. It's amazing that this hasn't happened so far. And he traces it back to Egypt, yes. So mummies. And I think they're mastectomies. So there must have been a doctor who thought about cutting out a cancer, in a breast cancer. Yeah. Yeah.
Working on that, has it gotten worse? I mean, we hear about clusters.
Yes, yeah. So the best predictor of, or the best indicator that has gotten much worse is our lifespan. It's very clear. So our lifespan has increased so much that's why we have the problem, essentially. Yes.
Is it because the immune system depletes as you get older?
That is an interesting question. I think it is still not decisively clear. Yes. Yeah.
All right.
But you're good. You're doing well.
Thank you, Dr. Lloyd Trotman, Professor Lloyd Trotman.
Thank you. Thanks, everybody. Really cool.
Everybody, please buy a beer.